Okay, let me just double check the mic. Hello, 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 hello. Nothing so far. Did I actually turn this on? Hello, hello, hello. Yeah, I did. Nothing so far. Ah, here we go. Cool. Okay, cool. Um, so just gonna start with a bit of Greek. I'm not gonna be on for too long. I'm getting my vaccination tomorrow, so for COVID-19. Hooray! So just gonna be on for a little while. Right. Okay. Let's do this.
Me.
Me, which is the lowercase one.
Zo.
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 Zo. Guess it's that. So that is a Z. Cool.
Ze.
Z. Epsilon. Is that what it is? No, is that Omega? I think it's Omega. Select the correct characters for Z.
Ze. Z.
Yes, the rice. Yes was. I have a funny feeling it's that, but then I would be surprised if they got Z's in there. Interesting. Okay. Well, we didn't have that choice here, so that is pretty interesting. I still think
Ne.
it's that one.
To.
What's this, rice? So it's P was, is it Rosie or something like that?
Rizi.
Rezi, Rezi. This is I, Rezi, Rezi. Let me see if I can say that again.
Rizi.
Rezi. Interesting. Okay. Maybe I should report that as well. The dictionary hence on hover are wrong. Write this in Greek. The pink carrot.
To.
To. Pink. So this is rice, so this must be
Rose.
pink. Rose. Ah, that makes sense. Rose. Carotto.
Caroto.
Carotto. Cool. Nice little start. Right, that's enough of that. So, what I want to do is just go through these tabs. I'm going to remind myself of everything. I'm going to actually read through the same document again because there's a couple of things that I don't quite remember. Things like what happens when a virus comes and attacks and then leaves a bit of its memory. I want to remember that stuff. But what I'm going to do is just read over it at a normal pace now. Um, not stress about all the names and stuff. And then, I mean, depending on the time, I think I'm gonna leave at 12. Um, I'm probably going to just take a little peek at these, probably worth remembering things, um, but not go too much into detail. This is literally just a revision of what I did on Wednesday, so that's what I'm going to do. This is decaf, by the way. I quite like the taste though. Okay, cool. So, number one, this is not what I wanted to do. That's not what I wanted to see. So, prokaryotes are unicellular organisms that lack organelles or other internal membrane-bound structures. Therefore, they do not have a nucleus, but instead generally have a single chromosome, a piece of circular, double-stranded DNA located in an area of the cell called the nucleoid. Prokaryotes. Okay, zinc finger. That is one of the ways of um, doing what CRISPR does now. I think it was. A zinc finger is a small protein structural motif that is characterized by the coordination of one or more zinc ions. In order to stabilize the fold, it was originally coined to describe the finger-like appearance of a hypothesized structure from the African clawed frog. However, it has been found to encompass a wide variety of differing protein structures in eukaryotic cells. Xenopus laevis, which is that frog, I guess, yep, was originally demonstrated to contain zinc and require the metal for function in 1983. Let me just search for DNA in here. DNA, nope. Okay. Maybe CRISPR, nope. First generation. Okay, that's not what I wanted to know. Fine, close. Eukaryote. A eukaryote are organisms whose cell have a nucleus. So prokaryote is without, and eukaryote is with. Okay. Uh, so whose cells have a nucleus enclosed with a nucleus envelope. Eukaryotes belong to the domain Eukaryota or Eukarya. This is, yeah, Greek. Their name comes from the Greek well or good. And caryon, there you go, caryon, nut or kernel. Okay, cool, we know that one now. How do you say, so, archaea, that's the one.
archaea,
Archaea. Streptococcus pyogenes was one of the main, hmm, one of the main somethings in CRISPR. Uh, nope, can't remember, but it is a Streptococcus is that virus thing, isn't it? Protospacer adjacent motif is when you have the helix. Wait, let me just draw this. This is just for revision purposes, by the way, so this might be wrong. I'm trying to remember, see if I can lodge it into my brain a bit. And you can't see any of this. Okay, cool. So I think it was, if you have this DNA helices, or whatever it's called, and you break it with CRISPR, there is PAM here and something else here, and I can't remember what that was. And I can't remember if you need it or it's just there. So let's see, protospacer. So PAM for short is a short DNA sequence, usually 2 to 6 base pairs in length, that follows the DNA region targeted for cleavage. Huh. Hey, I wasn't too far off. Cool. Next one. The same one. This is the same. Base pair. The base pair is the, I think it's these parts. So when you have like a DNA that looks like that. Base pair. I think it's like, is it like this part or both parts here? Like that sort of thing. So let's see. A fundamental unit of double-stranded nucleic acids. So okay, so these are the nucleic acids. Double-stranded. I think it must mean this then. So this is a base pair. Unit of double-stranded nucleic acids consisting of two nucleobases bound to each other by hydrogen bonds. They form the building blocks of the DNA double helix and contribute to the folded structure of both DNA and RNA. Dictated by specific hydrogen bonding patterns, Watson-Crick base pairs allow the DNA helix to maintain a regular helical structure that is subtly dependent on its nucleotide sequence. The complementary nature of this base pair structure provides a redundant copy of the genetic information encoded in each strand of the DNA. Well, we knew that from last time, so yeah, it has to represent this then. I'd love to see a picture actually understand. Depiction of the adenine diamine Watson-Crick base pair. These are interesting. I don't know what they mean. Let's see if there's any explanation in here. No. Let's see what this one is. Top, a GC base pair with 3 hydrogen bonds. Bottom, an AT base pair with 2 hydrogen bonds. 3 hydrogen bonds. Let's see. Hmm. I'm looking forward to knowing what these mean. I can't quite figure it out at the moment. But okay, so that's interesting. The base pair seems to be obviously more complex than this kind of illustration that people tend to use. So I guess this is literally either this or it won't be that. It'll be this. I think that's right. We'll come back to it anyway. RNA polymerase. So RNA is the so I know it from COVID-19 because the RNA is the thing that um thing that goes off to create copies of itself, or it's like the programming of it. But then there's the guide RNA, which is the thing that guides the cleavage of the DNA. So let's just actually read the actual answer now. In molecular biology, RNA polymerase, abbreviated RNAP or RNA pol, an officially DNA-directed RNA polymerase, is an enzyme that synthesizes RNA from a DNA template. Using the enzyme helicase, we know this stuff, okay. RNAP locally opens the double-stranded DNA so that one strand of the exposed nucleotides can be used as a template for... Just cut off. How strange. I think I'm still streaming. Hello, hello, hello. I'm still streaming? Yeah, okay, well, sorry about that. So, we're talking about RNA polymerase, which is an enzyme, okay, and it's a helicase enzyme. No, using the enzyme helicase, yeah, okay. So we have this helix, and we cut here, and it says locally opens the double-stranded DNA so that one strand of the exposed nucleotides can be used as a template for the synthesis of RNA. So when this breaks up, this is now loose, and it in itself creates RNA. And the thing that creates it is no, helicase is the thing that breaks it. I think we've had this diagram before actually, but helicase, and then the polymerase locally opens the double strand DNA so that one strand of the exposed nucleotides can be used as template for the synthesis of RNA. Okay, fine. Transcription factor. Now this was how much data can be delivered to the RNA to DNA transition. So I remember the diagram. Once again, I'm going to draw this helix. I don't think anyone's watching, but if they are, this is simply me just trying to remember what I learned. Yeah, so transcription factor is when it's divided up, there is two things that are really important to have at the end. One of them's a transcription factor, and then the other one was, and I can't remember it, so we're going to have a look at it. It's one of these here somewhere. Here, transcription factor and a mediator complex. Cool. Mediator complex. I'll probably forget that one again. A transcription factor or sequence-specific DNA binding factor is a protein that controls the rate of transcription of genetic information from DNA to messenger RNA. Ah, so it's the other way around. Cool. DNA to RNA. So it's the RNA that gets generated by the polymerase, the what's the word for that? Was it RNA polymerase? Yes, RNAP. That thing. Um, so transcription factor is a protein that controls the rate of transcription of genetic information from DNA to messenger RNA. So when that pops off, let's say um, this RNA's just come off, generated by this. Um, the transcription factor is determining how much data gets delivered from the DNA to the RNA. That makes sense. Okay. Nucleo means relating to or of the nucleus, I think. Let's see. Formed from nucleus. Oh no, this is the word, sorry. Forming words pertaining to nuclei. Okay. Nuclei. Mer was polymer. Damn, I can't remember. Used to form words relating to chemical structure, denoting parts of a molecule. For example, monomer, one part. Dimer, two parts. It's a part. Okay, mer is a part. That's really good to know. I don't know. I hope I don't forget that one. Proto is, I think it's before. Can't quite remember. Let's have a look. Proto. First, yeah. Most recent common ancestor. Okay. Oh, what was a protospacer though? I didn't actually. Ha! I know this one. So let's go protospacer. A protospacer adjacent motif, PAM, is a 2 to 6 base pair DNA. Interesting. So 2 to 6 base pairs. A sequence immediately following the DNA sequence targeted by the Cas9 nuclease. The helicase thing, isn't it? Or no, Cas9 targeted by the Cas9 nuclease. Maybe. In the CRISPR bacterial adaptive immune system. The PAM is a component of the invading virus or plasmid, but is not found in the bacterial host genome and hence is not a component of bacterial CRISPR locus. Protospacer is a 2 to 6 base pair DNA sequence immediately following the DNA sequence targeted by the Cas9. Okay, cool. Caryo, before vowels carry word forming element used since nut. Okay, that's Karyan. Yeah, okay. Bacteriophages, etymology. It's funny because I think I've forgotten most of what I've just read, but at the time I feel like I've absorbed it. But anyway, bacteriophage is that's the etymology. What the hell is this? Type of virus that infects bacteria. So what does phage mean? Oh, excuse me. Phage. Eat. Okay. Ribose. Ribose was a sugar, I think. Naturally occurring pentose sugar. Component of the nucleosides and nucleotides that constitute the nucleic acid bipolymer RNA. It is also found in riboflavin. So, I need to read that again. A naturally occurring pentose sugar, which is a component of the nucleosides and nucleotides that constitute the nucleic acid bipolymer RNA. That's a heavy sentence. We'll come back to it. Duplex etymology. So duplex etymology, that one. Duplex means two, yeah, cool. Annealed means like bound together with heat. Oh yeah, it is heat, yeah. Is a heat treatment process used mostly to increase the ductility and reduce the hardness of a material. Okay. Nucleotide DNA. Yeah, the nucleotides are those little segments. Yeah. archaea. So archaea was the one I just read out, and it is something to do with being like single-celled. I think it's single cell or a group of them. Microorganisms which are similar to bacteria in size and simplicity of structure, but radically different in molecular organisms. Okay, so a domain of single-celled organisms. These microorganisms lack cell nuclei and are therefore prokaryotes. And then eukaryotes are with, prokaryotes are without. This makes a lot of sense. Homologous means, I can't remember. Having the same relation relative. Oh yeah, that's right. It talks about the arm, doesn't it? A seal's flipper is homologous with the human arm. I should know that because I'm a developer. Shame. Homology. Okay. Codons. I don't remember that. That was near the end. A codon is a trinucleotide. So these trinucleotide sequence of DNA or RNA that corresponds to a specific amino acid. So these nucleotides are these things. These are nucleotides, these things. A trinucleotide would be three. I wonder, so I remember reading that if something isn't divisible by three, then it will cause mutation to occur. But I don't think that's what that's saying. So is a trinucleotide sequence of DNA or RNA that corresponds to a specific amino acid. The genetic code describes the relationship between the sequence of DNA bases, A, C, G, and T, in a gene and the corresponding protein sequence that it encodes. The cell reads the sequence of the gene in groups of three bases. Fine. I don't know what that is. After reading that. Can you see this? I think you can actually. I think you can see this. Um, Phenotype. So what does that mean? I can't quite remember. Pheno. The sum total of the observable characteristics of an individual, which is a type of organism distinguishable from others by observable features. So something has a phenotype. The noun phenotype entered the lexicon around 1910, adapted from the German word. Okay, German. Oh, okay. Based on the Greek pheno, which means shining or appearance. That's interesting. Sum of total observable characteristics of an individual phenotype. All right. So that was good. Um, I think we should still read this one as well. So I think um, I'm going to take some good time over this because this is the absolute foundation of my knowledge here. I need to make sure I understand it. So the idea was to literally just read over this entire document again, um, with less stops to try and understand and see how much sinks in essentially. So here we go. Genome editing involves the deletion, insertion, or modification of specific DNA sequences in the genome. For many years, researchers had been trying to develop easy and cost-effective genome editing tools to address problems across a wide spectrum of fields. For instance, gene therapy in humans could progress rapidly if one could simply eliminate the gene responsible for a certain genetic disorder. In agriculture, manipulating plant DNA could be used to optimize crop yields and control plant diseases. Let me just check the audio. Hello, hello, hello. Cool. Similarly, bacterial genomes could be fine-tuned to increase their product yields in several industrial applications. Finally, the efforts of researchers paid off with the development of CRISPR, a robust molecular tool that can edit DNA at virtually any locus. CRISPR technology is igniting a revolution across the life sciences and blah blah blah. Tools before CRISPR. Although CRISPR has now become synonymous with gene editing, it is not the first technology developed to edit DNA. Genome editing techniques initially emerged with the discovery of restriction enzymes and meganucleases. The possibility of precision gene editing was made clearer with the discovery of zinc finger nucleases. The ZFN method involves engineering an enzyme with both a zinc finger DNA binding domain and a restriction endonuclease domain. The zinc finger domain is composed of 3 base pair site on DNA designed to target and bind to specific sequences of DNA, and the nuclease domain cleaves the DNA at the desired site. Although zinc finger editing represented the first breakthrough in site-specific genome engineering, they have several limitations. In addition to exhibiting off-target effects, zinc fingers are expensive and time-consuming to engineer. Furthermore, their inefficiency limits their practical application to only genomic edit at a time, only one genomic edit at a time. Many years after zinc fingers made their debut, a similar method known as transcription activator-like effector nucleases, Talons, was developed. The Talons method utilizes engineered enzymes containing a DNA binding domain and a separate DNA cleaving domain, similar to the zinc fingers method. However, Talons have an advantage over zinc fingers because they are more flexible. Their DNA binding domains can target a wider range of sequences, although they are easier to design than zinc fingers. Talons are expensive to produce. That's interesting, just to say that I think I understand this now. It says separate process for the binding and separation. Yeah, so the Talons method utilizes engineered enzymes containing a DNA binding domain, which would be the kind of, I guess it would all be in one process in CRISPR, and a separate DNA cleaving domain. So the Cas9 comes in, so this would be the Cas9 part of CRISPR, and this would be like the guide RNA guiding it into it. An additional genome editing technique uses engineered restriction enzymes in concert with recombinant adeno-associated viruses, RAVs. AAV, or adeno-associated viruses, is a non-pathogenic virus that infects mammalian cells at all stages of the cell cycle and integrates into the host genome at predictable sites. The adeno-associated virus. The adeno-associated virus's genome can be modified to target specific sequences in the host genome and integrate desired DNA sequences. Adeno-associated. I don't know why I keep forgetting it. Terrible memory. However, the adeno-associated virus's approach also has several limitations. For instance, the vectors are difficult to produce and can only accommodate a small amount of genetic material. Adeno-associated virus. Up until now, the field of genome engineering has provided researchers with a few gene editing technologies, all of which had limitations. Table 1. Let's have a look at Table 1. Cost is high for zinc fingers, high for Talons, moderate for adeno-activated viruses, and low for CRISPR. The complexity of zinc fingers, Talons, and adeno-activated viruses, difficult, easy for CRISPR. Multiple edits, all difficult. Zinc fingers, Talons, can't remember this one. This was transcription activator-like effector nucleases. Transcription activator-like engineered effector nucleases. A adeno-activated virus. Okay, cool. So because zinc fingers and Talons require complex protein DNA interactions, Talons being God, activated like, no, something like effector nucleases. T is transcription activator like effector nucleases. I'm going to forget that. AAV, adeno-activated virus, transcription activated, no, it was transcription activator-like effector nucleases. Okay, CRISPR is a simple two-component system that can be used to target a specific genomic sequence and then make a cut in the DNA. This powerful tool enables scientists to manipulate a specific gene of interest. For instance, one may knock out a gene, making it inactive, or knock in a particular sequence that repairs a mutation or adds a characteristic. Below, we describe how CRISPR was discovered and developed into a powerful genome editing tool. Okay, cool. This is transcription activator-like effector nucleases, adeno-activated virus, zinc fingers. Gotcha. The foundational discoveries that led to the development of CRISPR Cas9 technology can be traced back to 1993 when repetitive palindromic segments of DNA interspaced with other fragments of genetic material were identified in prokaryotes. This is pro, so it's without a nuclei, whatever it's called. These repeated segments of genetic code were named clustered regularly interspaced short palindromic repeats. Or clustered regularly interspaced short palindromic repeats. However, the segments of DNA between the repeats were really the interesting part. Clustered regularly interspaced short palindromic repeats. I'm not going to remember that. In 2007, after years of study, researchers concluded that clustered regularly interspaced short. Let me read it again. In 2007, after years of study, researchers concluded that clustered regularly interspaced short palindromic repeats function is related to prokaryotic immunity. It took the combined efforts of several research groups over the next 5 years to elucidate the underlying molecular mechanism behind the clustered something interspaced short palindromic repeats. That one is clustered regularly. As it turns out, bacteria and archaea is a single cell, use the clustered what's it called again? Fuck, I'm gonna remember that. Clustered regularly interspaced short palindromic repeats, Cas9 system. So okay, my apologies, let me just read it again. As it turns out, bacteria and archaea use the clustered regularly interspaced short palindromic repeats, Cas9 system, to defend themselves against invading viruses called bacteriophages. Upon encountering a viral infection, the prokaryotic cell employs a special clustered regularly interspaced short palindromic repeat associated nuclease, Cas9, to snip off a piece of viral DNA by creating a double-strand break in its target loci. How does the Cas9 protein recognize the target DNA? It is directed to the target sequence by a short RNA fragment known as a guide RNA. The guide RNA is complementary to a segment of the viral genome, which allows Cas9 to cleave DNA with a high degree of specificity. Not only does cleaving the DNA destroy the virus, but a fragment of foreign DNA called a spacer may be stored between the palindromic sequences of the CRISPR array as a way of retaining a genetic memory of past infections. If the virus were to re-invade, the CRISPR-Cas9 system could quickly target and destroy it. This library of viral fragments is thus essentially equivalent to our immune system, which stores antigens to prepare for future infections. Once scientists figured out the mechanism of clustered, regularly interspaced, short palindromic repeats in prokaryotes, it did not take long for them to realize the potential for engineering the genomes of microbes, plants, and animals. Today, clustered, regularly interspaced, short palindromic repeats is utilized for a variety of applications, and its adoption continues to increase in laboratories throughout the world. Adeno-activated virus, transcription activator, what is it? What is this one? Activator like, haha. Transcription activator like effector nucleases. Okay. CRISPR, a set of DNA sequences involved in the prokaryotic immune system that has been adapted for genome engineering. The components of CRISPR. The CRISPR system comprises two components, a guide RNA that is specific to the target DNA sequence, and a clustered, regularly interspaced, short palindromic repeat associated endonuclease, Cas. Okay, let me just read that again. The CRISPR system comprises two components, a guide RNA that is specific to the target DNA sequence, and a CRISPR-associated endonuclease, Cas. For CRISPR experiments, the guide RNA and Cas endonuclease are combined to form a ribonucleoprotein, or RNP, complex. The Cas protein functions as a pair of molecular scissors, while the guide RNA is the GPS that guides it to the appropriate site. In prokaryotes, the guide RNA guides the nuclease to viral DNA, but as a biotechnological tool, the design specifications of the guide RNA can be altered to target any organism's genome at virtually any location. For many genome engineering applications, the Cas9 protein from Streptococcus pyogenes is used. Binding of the guide RNA to the genomic target is contingent upon the presence of a short protospace adjacent motif, PAM, located directly downstream of the target on the opposite DNA strand. Go and check figure 1. So for many genome engineering applications, the Cas9 protein, Streptococcus pyogenes, is used. Binding of the guide RNA to the genomic target is contingent upon the presence of a short protospace adjacent motif, PAM, here. So it absolutely requires this. And it also needs this other one, NCC. I don't think we ever realized what that was. The Cas proteins from different prokaryotic species recognize different what is that one again? Protospace adjacent motif. The Cas proteins from different prokaryotic species recognize different protospacer adjacent motifs. The protospacer adjacent motif for Cas9 is 5, this one here, to NGG, I don't see that anywhere, to 3. So it goes, woo, there it is, NGG, okay, 3 here, cool. If a correct protospacer adjacent motif match is made, and if the guide RNA successfully binds to the target, then Cas9 cleaves both DNA strands 3 to 4 nucleotides upstream of the PAM site, the protospacer adjacent motif. In nature, this short genetic element only occurs in invading viruses, not the bacterial genome, and thus ensures that Cas9 does not cleave its own clustered, regularly interspaced short prokaryotic repeat locus. So this figure 1, the CRISPR-Cas9 system comprises a guide RNA and a Cas9 nuclease, which together form a ribonucleoprotein complex. The presence of a specific protospace adjacent motif in the genomic DNA is required for the guide RNA to target to the presence of a specific protospace adjacent motif in the genomic DNA is required for the guide RNA to bind to the target sequence. The Cas9 nuclease then makes a double strand break in the DNA, denoted by the scissors. Endogenous repair mechanisms triggered by the double strand break may result in gene knockout via a frame shift mutation or knock-in of a desired sequence of a DNA template is present. Now this frame shift mutation is that not divisible by 3 thing. So that's interesting. So as a summary, the guide RNA is the RNA component of the CRISPR-Cas9 genome engineering tool. It contains a roughly 20 base pair sequence that is complementary to the genomic target. So it's added to it. CRISPR-associated endonuclease protein, Cas. Let me just read this one again. It says clustered, regularly interspaced short prokaryotic repeater associated endonuclease protein, a family of proteins that is able to cleave nucleic acids. Cas9 is the most commonly used Cas protein in CRISPR experiments. So I'm going to come back to this because this actually gives me the information I need. I need to have the endonuclease protein to cleave the nucleic acid. And so what I want to do is I'm going to buy a pipetting, like a manual one first. And I'm going to just break down some DNA and take a look. So I need to have a nice magnifying glass, microscope. Yeah, and some way to apply the proteins. And the protospace adjacent motif, a short sequence of nucleotides that must be present downstream of the target site in order for the nuclease to make a double strand break. It'll be interesting to see what that actually looks like in real life. It seems all a bit abstract at the moment. So guide RNA formats. In its natural form, the guide RNA consists of 2 distinct segments of RNA, CRISPR RNA and transactivating CRISPR RNA, tracer RNA. The CRISPR RNA is an 18 to 20 base pair sequence that binds the genomic target. The tracer RNA functions as a scaffold for the CRISPR RNA Cas9 interaction. In natural context, guide RNAs form a duplex molecule with the CRISPR RNA and tracer RNA segments annealed together, denoted as CR ratio thingy colon tracer RNA. However, guide RNAs can now be synthetically produced with CRISPR RNA and tracer RNA connected by a linker loop, that blue thing. These seamless molecules are called a single guide RNAs. Okay. I feel like I've really accelerated through this. I feel like I'm probably annoying most people with my voice as well. Luckily no one's watching, that's great. Okay, so 2-piece RNA. And then the single guide RNA uses this linker loop. The 2-piece and single guide RNA formats. Guide RNAs can be constructed in 2 formats. The CRISPR RNA green and tracer RNA purple. Components may be annealed together to form a 2-piece guide. They are annealed together with the linker loop. They may be connected by a linker loop to form a continuous molecule, single guide RNA or single guide RNA. Okay. Creating double strand breaks. For many CRISPR-Cas experiments, Cas9 facilitates the gene editing process by let me just remember what this one is again. It is here somewhere. Cas is CRISPR-associated endonuclease. Okay, I don't think I remember that. So it's clustered, regularly interspaced, short, prokaryotic repeater, clustered, associated. I didn't think I was going to remember it. CRISPR-associated endonuclease. For many CRISPR-Cas experiments, Cas9 facilitates the gene editing process by creating a DSB, a double-strand break in the DNA, similar to what occurs in bacteria and archaea. Generating a double-strand break is the initial step in the CRISPR editing pathway. The repair mechanism that follows will dictate the type of gene editing that occurs and will influence your desired editing outcome. In the next section, we will explore 2 main types of repair: non-homologous end-joining, NHEJ, and homology-directed repair, HDR. Okay, so, excuse me. So we've got this thing. So it says 5 hyphen NGG hyphen 3. The cut requires the Protospacer adjacent motif. It needs to be there, and the linker loop binds together with a heat roughingness thingy. What was that called again? That is called the annealed. The linker loop is annealed together to the tracer, no, the CRISPR RNA and the tracer RNA. Woohoo! I got it right. Yep. Okay. And then that is a double strand break in this pair. This sequence of nucleic acids. Figure 3 CRISPR-Cas components create a double strand break. The single guide RNA and Cas9 complex forms a ribonucleoprotein. Sugar. Ribose. I swear that was the sugar. The sgRNA bind. No, it can't be. Was that really the sugar? Ribonucleoprotein. Ribonucleoprotein. Sugar. Sugar phosphate back. Maybe not. That's a bit crap. Let's have a look at this. Ah, you know what? It was etymology, I think. Etymology. I just want to double check this. Ribose. Okay, so maybe not the same. Pentose, sugar. It could be. It could be the same. Ribonucleoprotein. The single guide RNA binds the genomic target, and Cas9 makes a CRISPR associated. Sure, I'm not gonna ever remember these damn things. CRISPR associated endonuclease. Of course, S. That makes a lot of sense. Guess otherwise it would be called CAN. So the single guide RNA and CRISPR associated endonuclease 9 complex forms a ribonucleoprotein. The single guide RNA binds to the genomic target, and Cas9 makes a double strand break in the DNA. Repairing CRISPR-induced breaks. As described above, CRISPR facilitates the generation of double strand breaks at specific locations in the genome. However, this action alone does not cause a gene to be edited. Rather, the process of repairing the break enables changes to be made to the target gene. After a double strand break is made, innate DNA repair mechanisms are automatically triggered within the cell. Below we'll discuss two main types of repair pathways that are often used to edit genes: non-homologous end joining and homology-directed repair. The non-homologous end joining, if the experimental objective is to permanently disrupt the gene so that no functional protein is made, a knockout, then the non-homologous end joining repair mechanisms can be exploited. Figure 4 on the left. Non-homologous end joining repair mechanism. You insert it as a replacement, I think it was, wasn't it? Or deletion. I swear the insertion was like a direct replacement of what should have been there. Non-homologous end joining ligates the DNA ends back together, but it is prone to error and may insert or delete nucleotides called indels. In the process, if the number of nucleotides inserted is not divisible by 3, then it will induce a frameshift mutation and likely terminate the resulting protein's function. Homology-directed repair. If the objective of the experiment is to replace the targeted genetic element with a different sequence, a knock-in, the cell can be directed towards the homology-directed repair. A DNA donor template bearing the desired sequence flanked by regions of homology must be introduced along with the CRISPR components to the cells. The cells will use this template to repair the broken sequence via homologous recombination. thereby incorporating the desired changes into the target region. Figure 4 on the right. So the double strand break, this is homology arms. We just talked about the knock-in sequence goes in. This is going to modify it, and this is homology directed repair, where we've got like something similar, but it's different. Like the fin of a penguin is the same as a human arm. That makes sense. So figure 4, CRISPR repair mechanisms. The two most common repair mechanisms facilitating CRISPR-Cas editing are non-homologous end joining and homology-directed repair. Non-homology end joining results in either insertions or deletions of nucleotides to repair the double-strand break. HUDR repairs the DSB by incorporating a DNA donor template into the target sequence. HDR is homology-directed repair. Indels, the insertion of or deletion of nucleotides in the genome, often caused by non-homologous end joining following a double-strand break in DNA. DNA donor template, a specified DNA sequence desired to be knocked into the genome that is flanked by homology arms to facilitate homologous recombination. So the the homologous end joining is where you cut, replace directly, or cut and delete. It's error prone and can cause the frame shift mutations. The homology directed repair is where you place something in, like a replacement, a homology, or something that is similar to what was there before but different, and that was called a repairs by incorporating a DNA donor template into the target sequence. Donor template. Cool. Components and mechanisms of CRISPR-Cas9 editing. Components of CRISPR-Cas9. So Cas9, the single guide RNA, the CRISPR RNA, the tracer RNA, the linker loop, that's amylase. No, amylase. No, it was not that. It was up here. It was definitely here somewhere. Damn, where is it? Annealed together. Annealed. So the linker loop is annealed. It is annealed to the CRISPR RNA and tracer RNA. The guide RNA target sequence is this 20 base pair strand of nucleotides, or the CRISPR RNA to trace RNA. I actually can't remember what that stood for, but I remember this thing. 20 nucleotides. Okay, cool. So the composite CRISPR-9, 1, 2, the RNP formation. RNP is the ribonucleo protein. Ribonucleotide or nucleic? Ribonucleic protein or something. Ribonucleoprotein. So there we are. So this is a ribonucleoprotein. Okay, and the protein is used to break it up. This is the snipping, the cleavage. So this leads to the Cas9 mediated double strand break. The double strand break is now entered into the drawing. It requires the associated motif, the prokaryotic associated motif. Okay, double strand break occurs, then you can either replace it with a non-homologous end joining, this one repair, which is error prone, or deletion, or you can use the homology directed repair, using the homology arms knock-in sequence. It's a template um, replacement. Figure 5. CRISPR-Cas9 genome editing. The components of CRISPR are a guide RNA, either single guide RNA or CRISPR tracer, and a Cas9 endonuclease. 2. The guide RNA and Cas9 form a ribonucleoprotein. 3. Inside the cell, the RNP creates a double-strand break at the genomic target. One of two endogenous repair mechanisms may mend the break. 4. A. Non-homologous end joining is an error-prone pathway that often inserts or deletes nucleotides. Indels. 4b. If a DNA template is provided, then the homology-directed repair pathway may mend the break through homologous recombination. Okay, this is good. I feel like it's slowly going in, very slowly going in. What can we achieve using CRISPR? Once scientists realize the potential of CRISPR, the field of genome, blah blah blah. Gene tagging. This is using um, lit. Fluorescent genes. Screening, anti-CRISPR, CRISPR-I, knock-ins and knockouts. Okay. So making a gene permanently inoperative does not encode a functional protein is called a knockout. CRISPR's ability to disrupt gene function relies on the error-prone nature of the non-homologous end joining mechanism. As described above, indels that cause shifts in the reading frame of a gene will terminate the gene's function. This is especially true for frame shifts that cause premature stop codons. Yeah, I don't remember that one. A knockout can also be achieved using multi-guide single guide RNA, multiple guide RNAs that target the same gene. By making multiple simultaneous cuts in the DNA, the single guide RNAs induce one or more large fragment deletions in target genes. Because these deletions remove several amino acids, they often render the targeted gene completely inoperative. Synthego's gene knockout kit V2 and screening libraries utilize this multi-guide approach to robustly knock out genes. By purposefully disrupting the sequence of a gene and corresponding mRNA and protein, researchers can elucidate the impact of the knockout of the phenotype of a cell or organism. This technique is thus useful for a variety of applications, including the identification and validation of potential drug targets, the analysis of cellular pathways, and the validation of antibodies. So knock-ins, we know what they are. They are the replacement templates um, that go in after the homology-directed repair. The incorporation of genetic material into a cell's genome is referred to as a knock-in. These edits are achieved by inducing cells to repair dual-strand breaks through homology-directed repair. For homology-directed repair experiments, a DNA template containing a knock-in sequence flanked by regions of homology must be introduced into cells along with the CRISPR components. This is CRISPR is it's like what is it? Grunt CRISPR. This is something like chunked. Oh damn! I really forgotten it all. This is combined, compressed, something regularly interspaced, short prokaryotic repeater. What is that? Clustered regularly interspaced short prokaryotic repeater components. The template is used to precisely repair the severed target sequence, incorporating the knock-in sequence in the process. Homology-directed repair enables countless genomic rewriting applications, from introducing single-point mutations to inserting selectable markers. While the homology-directed repair technique requires further refinement, researchers have already employed the method to correct a genetically encoded mutation causing cataracts in mice, demonstrating proof of concept for CRISPR. While gene knockouts can be, so I'm going to slow down a bit because I don't remember this stuff. While gene knockouts can be achieved by disrupting a genetic sequence, gene expression can be repressed without altering the corresponding DNA. In 2013, QI et al. Made a Cas9 variant by mutating the endonuclease domains so that the enzyme no longer cuts DNA. Hello, yes, it's working. No longer cuts DNA, called dead Cas or dCas9. Researchers have used dCas9 to develop a technique wherein the CRISPR complex binds to its DNA target but does not cleave it. The binding of the dCas9 prevents the cell's transcription machinery from accessing the promoter, thereby inhibiting the gene's expression. Fusing a transcriptional repressor domain such as CRAB to dCas9 allows reversible and fine-tuned reduction in gene expression. Figure 7a. Yeah, dCas9, an inhibitor, the gene is silenced. Okay, promoter as well. Oh, excuse me. Oh. In a nod to the precursor gene silencing technique, RNA interference or RNAi, the CRISPR silencing technique has been termed CRISPR interference or CRISPR, whereas RNAi interference represses genes by destroying RNA transcripts. CRISPR-I represses genes at the DNA level. Compared to RNAi, CRISPR-I is associated with higher efficiency, greater versatility, and fewer off-target effects. Don't like this. This word. CRISPR activation. While dCas9 endonucleases can be used to silence gene expression, they can also be modified to activate target genes called CRISPR activation or CRISPR-A. By fusing a transcriptional activator such as VP64 or VP16 to dCas9, scientists are now developing systems that can overexpress genes of interest. Polstein and Gersbach created one such system in 2015 by fusing light-inducible proteins to mutated Cas9, causing gene expression to be activated in the presence of blue light and repressed in its absence. Other teams of researchers such as Zalatan et al. 2015 are building more complex systems for multiplex gene activation and repression at as many as 3 loci simultaneously. Figure 7, mechanisms of CRISPR interference and activation. So the top one was the spacers at the bottom, then you've got the interrupter, I think that's it, and then the gene expressed comes out, or gene silence, that's the one, inhibitor, okay. CRISPR activation are used catalytically dead Cas9 to alter the gene expression of targets without changing the genetic sequence. Depending on the transcriptional regulator used, gene expression can be A, inhibited, or B, activated. CRISPR screens. Another application of CRISPR is genome-wide functional screening. Until recently, RNAi interrupted was the primary approach for loss ribonucleotide activator interrupter. RNA, let me just double check that. What does RNA stand for? I think it is that. Ribonucleic acid, okay. So ribonucleic acid interrupter was the primary approach for loss of function screens, where genes are systematically inhibited across the genome to determine their function. However, as mentioned previously, RNAi is plagued with problems related to low efficiency and high off-target effects. Now with CRISPR, guide RNA libraries are being developed to knock out hundreds of genes in a single screen with high efficiency. Synthego now offers several arrayed single guide RNA libraries that target one gene per well of a multi-well plate. These screens can be used for various applications, including the identification of potential drug targets. So you've got your Petri dish, you've got your Cas9, using the guide RNA and the CRISPR, this is the CRISPR library. You plop this into the well, systematically disrupt a set of genes using a CRISPR library. I guess the well is filled with genes, and this mother is gonna go. Oh, actually, okay, that's interesting. Apply drug or treatment. So you're applying this feature to this set of genes, faulty genes, I guess. So CRISPR-arrayed screening workflow. A guide RNA targeting each gene is introduced to each well of a multi-well plate. Applying a treatment such as a drug to cells enables one to identify which genes, when rendered inoperative, cause the cells to have heightened or diminished sensitivity to the drug. This process can be used to identify potential drug targets. Anti-CRISPR. Although the CRISPR-Cas9 system enables fine-tuning of genomic DNA, one downside is the risk of off-target effects, cutting DNA in the wrong place. One solution to this issue is to harness anti-CRISPR proteins that inhibit Cas9 activity. Cas9 is the thing that you guide with the RNA to break it up. So if you have anti-CRISPR proteins that inhibit Cas9 activity, that means they're not going to break. Anti-CRISPR, anti-cluster, clustered, regularly interspaced, short prokaryotic repeater proteins that inhabit. In nature, bacteriophages use these proteins to evade the CRISPR machinery of the prokaryotes. Poor luck. 2016 discovered anti-CRISPR protein inhibitors that were effective against Cas9 nuclease from Neisseria meningitis. In fact, the team showed inhibition of the enemy Cas9 activity in bacterial and mammalian cells using these anti-CRISPR proteins. Ultimately, this technology can be used to reduce editor errors. It turns out that adding anti-CRISPR proteins after editing takes place only partially reduces cleavage at on-target sites, but greatly reduces cleavage at off-target sites. I wonder how this works. Like, what's the mechanical part of it? You know, I think maybe we need to watch some videos of using CRISPR. I'd like to know, and I still like to know the actual ingredients I need to get, the materials I need to get to do this in my own dish. I'm going to be moving hopefully soon, and I think I'll buy all this stuff for that as like a celebration so I can set up my little lab and start doing some actual physical work. And nearly at the end of this, so gene visualizations. The CRISPR system can also be used to visualize genomic regions. This is achieved by attaching fluorescent proteins like green fluorescent protein GFP to dead Cas9 proteins or guide RNAs and using the CRISPR systems to tag desired parts of the genome. This technology now enables scientists to visualize the nucleic acids in real time, a process that previously was extremely difficult to perform. This is really interesting. I really do want to see these nucleic acids. I'd love to see one, and I wonder if that means that I need to use GFPs. Yeah, cool. Right, this is the last one I'm going to read, I think. No, that's it. That is it. I think I've read it. Cool. The rest is Synthego thing. This is a great document. I hope they realise that. This is just have been so useful. It's packed full of good information, and it's structured so well. Right. Okay. So I've done that reading. Now I wanted to read through these, or just take a look at them. And I think I'm gonna have to make a move because I need to be ready for my vaccination. So let me open that one up. Gene Knockout Kit, version 2. The most reliable knockout strategy for any human or mouse-derived cell lines. Ships in as few as 2 days. What's new with the Gene Knockout Kit? Updated pricing, human and mouse genomes now available. Ships in as few as 2 days. Let's see how much that costs. Wow, dollars. I wonder if they it won't be 2 days for me. Haha. Oh man. Okay. Oh, how long is this? Oh, this looks good though. This looks really, really good. Yeah. Wow. Oh, okay, okay, okay. I see the next episode coming on now. I think okay, so what I'm gonna do is I'm gonna probably rewatch this video. Um this literal video just to revise, and then next time I come on we'll go through this together. Anyway, so that's cool. So it looks like it's got some good stuff here. Let's see if I could just get a sniff of it. Multi-guide strategy consists of choose genes of interest, synthesize your high-quality multi-guide RNA kit. Concentrate your research is to reduce the number of clones we need to screen to find a desired target modification. Now test it. Okay. Powered by a unique multi-guide algorithm, the gene. Cool. That's nice. The gene knockout kit version 2 enables knockout of any human or mouse protein coding gene. Any human. To help you optimize your experiment, a transfection optimization kit that includes a control multi-guide single guide RNA and Cas9 human or a ROSA control single guide mouse are available. Okay, fine. Next. So screening libraries. Arrayed CRISPR screening libraries. Human or mouse CRISPR knockout libraries for confident target discovery ships next day. What is this about? This is okay. Obtain higher indel frequency with multi-guide sgRNA. Well, that's interesting. So indels, though, are they're the things that sounded like negative. They are part of the homologous, no, homologous end joining. I'm pretty sure that's right. Homologous end joining. Just search for indel. Yeah, non-homologous end joining ligates the DNA ends back together, but it is prone to error and may insert or delete nucleotides called indels. So better together, obtain higher indel frequency with multi-guide single guide RNA. I can't quite read that. Here we go. So it says so with these so these were mentioned in there. They were mentioned somewhere. dy was this here. dyrk4 rk4. Actually, no, maybe it wasn't. It was something else. It was mentioned in one of those little pictures, I think. It's here somewhere. I think it was 16 something. Okay. Oh, okay, they're different. VP64, VP16. Transcriptional activators. I don't know what those are. I hope it tells you somewhere. I'm trying to think. DYRK4 gene cause is a testis-specific kinase with a very restricted expression to stage V3 post-meiotic spermatids. Sorry, wrong word. Spermatids. I don't know what that is, so we'll have to figure that out. But the single-guide RNA 1, the indel frequency is. I mean, surely this is worse. Fragment deletion. Unless you're trying to destroy. Oh! Unless you're trying to destroy something. That might be what that is. Okay, well, look, clearly there's a lot of reading here to understand that, so that's cool. Right, good. Next one, Gene Knockout ebook. Oh yeah, that sounds good. I actually just gonna check this. Right, that's really cool. Um, Designing CRISPR knock-in experiments. Let me see what that is. So these are books, essentially. 2 books. Analysis of edits. Oh, okay. Let's have a look at that together. Synthego is the company. ICE analysis. Get NGS quality results with Sanger data in seconds. Download template, view example results. Efficiency. PRKG. GRK2. So these are clearly things I guess these are quite common things. You'll be like, oh, that's good. Look, guide target. Ha, that's cool. Prokaryotic adjacent motif. No, protospace adjacent motif sequence. Bad deletions or indel. I've misunderstood indel because this looks like. If the experiment objective is to permanently disrupt a gene so that no functional protein is made, then the non-homologous end joining repair mechanisms can be exploited. Ligates the DNA ends back together, but it is prone to error and may insert or delete nucleotides called indels in the process. If the number of nucleotides inserted is or deleted is not divisible by 3, then it will induce a frameshift mutation, likely to terminate the resulting protein's function. I wonder if it's saying. So the efficiency is saying maybe. I don't understand these graphs. Is indel good or not? And the knock-in score. I wonder what this is referring to. Is this how many times the nucleic acids are no longer divisible by 3, something like that. Okay, we'll have a figure out of that one. And how to perform a CRISPR experiment. Now this sounds legit. Let's have a look at this. Um, it's just another ebook. Cool. So I think the next session, um, it might be sooner than I thought because I actually quite enjoyed this. It might be on the weekend at some point, but maybe Wednesday. We'll see, I guess. It just depends how tired I get from work. And I'm going to read through all of these, I think. If they end up being quite big, then um, maybe. Yeah, we'll just figure it out on the day, I guess. Good, I like it. So, episode 4, that was like no notes essentially. Uh, we went through the CRISPR documentation again. We're going to read through the episode 3 probably worth remembering stuff in episode 5. Cool. That was fun. And perfectly timed. So have a good weekend and ciao for now.